This is the final portion of Chapter 13 on meiosis, and we left off with spermatogenesis and just briefly talking about how through the process of meiosis 1 and 2 we end up with 4 haploid gametes which develop into sperm. And one thing you should know about spermatogenesis is that it's a continuous and prolific process. By prolific we mean there are millions of sperm being produced on a continuous basis once maturation has occurred. And this is in contrast to the production of eggs. So egg production is called oogenesis, and it's similar in that it's going through the 2 meiotic divisions. But what's different about egg production, well, there are a couple things that are different, but one of the things that's different about it is that the onset of meiosis 2, instead of ending up with 2 very viable daughter cells that are the haploid daughter cells, we actually have one become a polar body. And there's actually some controversy about where the polar bodies are, at what stage they are produced. But essentially, the important thing that you need to know about egg production or oogenesis is that what we end up with, rather than 4 viable gametes at the end of meiosis 2, we only end up with 1. And we also end up with what are called polar bodies, which are different from the polar nuclei within the angiosperm egg. So these polar bodies actually, they're still learning about what function they serve. It was originally believed that they just kind of disappeared and were reabsorbed into the body, but there is now some thought that they perhaps help with some other functions within the body. But for what you need to know is at the end of meiosis 2, when we're talking about oogenesis, we end up with just 1 mature egg rather than 4 gametes. The other thing that is, and part of the reason that this is occurring, is that when we go through telophase 2 and telophase 1 and cytokinesis 1, we have during cytokinesis the division of the cytoplasm occurring, and in oogenesis that division of cytoplasm is not even. So we have an unequal division of the 2 cells, so that one of the cells ends up with more of the cytoplasm. And so then we have this occurring here. And so this polar body that's created in the first meiotic division doesn't go through meiosis 2 at all. The only cell that is going to go through meiosis 2 is this one viable cell. And so when this cell then divides in meiosis 2, it again is an uneven distribution of cytoplasm between the 2 as we go through cytokinesis, so that we end up with another polar body and a larger cell. So you can see the size difference between these 2. And this ends up being our haploid egg cell. Okay, so this is just a diagram showing you the same idea, but it's showing you where this is occurring here within the ovary of a female, and how we have these different portions of development occurring. And fertilization occurs within the fallopian tube. So the eggs are produced, and then during ovulation they're released, and then they are absorbed into the fallopian tube, which is where fertilization can occur. But these eggs are not occurring throughout a woman's life. Oops, we have to go back here. They're not occurring. In the male, we said it was a continuous process in terms of the production of gametes throughout life once maturation has occurred. But that's not the case with females, because these eggs actually are produced even before a woman is born. So before a female is born in utero is when these meiotic divisions are occurring within the ovary of the female. And these eggs just get stored within the woman and released one at a time, once a month, once maturation occurs. So, in general, that is the process that occurs. So, eggs are not being produced, new eggs are not produced. They are made at one time, and then that whole machinery just shuts down throughout the lifespan of a female. So going back to our summary of genetic variation, this is an animation that will just give us again a summary overview of how we get genetic variation through meiosis.
In sexually reproducing organisms, three processes lead to most genetic variation: independent orientation of chromosomes in meiosis, crossing over of chromosomes in meiosis, and random fertilization. Each pair of homologous chromosomes consists of one chromosome inherited from the father and one from the mother. Here we have color-coded them blue and red. Each pair of chromosomes lines up independently of the other pairs in metaphase 1 of meiosis. Here you see one of the possible arrangements and outcomes. There are two different ways that each chromosome pair can line up. That means that in the organism shown here, with the diploid number of 4, independent orientation of chromosomes at metaphase 1 can produce gametes with 4 different combinations of maternal and paternal chromosomes. In a human being, with 46 chromosomes, more than 8 million combinations are possible. Now let's look at how crossing over creates even more genetic variability. During prophase 1 of meiosis, homologous chromosomes pair up very closely, and corresponding parts of two non-sister chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from two parents. Here the process produced a total of 4 genetically different gametes. There are many ways crossing over can occur. In humans, crossover events happen an average of 2 or 3 times per chromosome pair, greatly increasing the variation among eggs and sperm. Note that crossing over produces some parental gametes with chromosomes like those of the parents, and some recombinant gametes with a mixture of genes from both sets of chromosomes. Independent orientation and crossing over occur simultaneously during meiosis, multiplying the number of genetic variations among gametes. Because each pair of chromosomes lines up independently, and crossovers can occur almost anywhere along each pair of chromosomes, it is possible for a human being to produce an almost infinite variety of gametes. A sperm fertilizes an egg, producing a zygote. The random nature of fertilization adds to the variation arising from meiosis. Each parent is capable of producing a huge variety of genetically different gametes. The number of possible combinations among their offspring is staggering. Theoretically, one human couple is capable of conceiving a number of genetically different offspring that is far greater than the number of humans who have ever lived.
So that's a pretty nice summary to again explain how we do have such a variety among all humans on Earth. And it's kind of nice. It makes you feel unique. So we're going to end up here with an overview of looking at the differences between mitosis and meiosis. So we don't want to confuse the two. And so there's a really nice chart here that goes over those differences. And I would pause and study this. And make sure that in your notes you have a very clear idea of the difference between the two. So this is nice because it breaks down. For example, with DNA replication, you can see in mitosis it occurs during interphase before mitosis even begins. And then in meiosis, it also occurs during interphase before meiosis 1 begins. So that's where our difference lies. With mitosis, we only have one division. With meiosis, we have two, and so on. So you can look through this. There are actually two slides here. This one also goes over, again, the differences between the two. You can see here chromosome replication occurring before prophase has started, and we end up with our sister chromatids. But in meiosis 1, you can see where we have tetrads being formed, and we do not have that occurring in mitosis. So again, I would pause this and study it and make sure you understand the differences and see if you're able to talk through what those differences are. And then this is just a very simple table that shouldn't have all these animations, but it does. Oops, we'll go back there to that. Just talking again with some really basic differences. So again, one daughter cell is genetically identical to the parent cell, or actually both two daughter cells. In mitosis, where the four daughter cells in meiosis are genetically different from the parent. So big difference right here between mitosis and meiosis. With mitosis, you end up with 2. With meiosis, you end up with 4. With mitosis, you go 2n to 2n. In meiosis, you go 2n to 1n. Mitosis, the whole purpose of it is to produce cells for growth or repair. There is no crossing over. Where in meiosis, the reason that we have it is to produce gametes. And there's, in humans, a very big difference of when the production of gametes occurs in males throughout life, adult life, and in females before birth. And again, in meiosis, there is crossing over. So finally, this is the credits. These PowerPoints are put together from Kim Foglia's Explore Biology website and from the Pearson Education from your Campbell textbook. Hope this helps.